The 22nd natural amino acid, well, natural for some archaea and bacteria, is not natural for most things, but we can use the machinery that these little buggers use to make this amino acid in order to incorporate unnatural amino acids, which allows us to like do really cool things like test the function of various parts of a protein in ways that are more site specific than and we can allow for more functionality, so expanding the possibilities of these like 20 canonical amino acids that we've been talking about. And we can do this by taking advantage of the machinery that these use to make this amino acid and then altering that machinery to get them to make something else. And so today I want to tell you about pyrrolysine, how it was discovered, how it's formed, and how we can take advantage of its machinery for unnatural amino acid incorporation in this like orthogonal manner. So proteins are like the workers of your cells, and they're made up of building blocks or letters called amino acids. And so as we've been going over this past month, so much more in past posts, but they have this same generic backbone where they have an amino group at one end and a carboxyl group at the other end, and then this unique side chain or R group. They link together through peptide bonds, and when they do this, only that like unique part, that R group, is going to like stick out, and this will give it, these side groups have these different properties, and these are going to influence how the protein is going to fold and function. The backbone is really important too, though, in order to give it like its secondary structure and things like that. So much more in past posts. But the basic idea is that these amino acids, the place where they differ is this side chain. And because these side chains have these different properties, things like differences in charge and hydrophobicity or hydrophilicity, so whether water likes to hang out with them or whether they have to kind of go together, like hide together in the center of a protein. So there's all sorts of different things, like some of these can be modified, various things after they're put into the protein. But we have these 20 basic canonical amino acids that our cells have to choose from, but they don't really get to choose. They're following the instructions that are the protein making instructions in that protein's messenger RNA, which is an RNA copy of the DNA. So the DNA gene has the like original recipe, but it has some extra regulatory information. And this is held up in the nucleus, and then in a process called transcription, your cells make RNA copies of this, and then those RNA copies get edited, that regulatory information removed and stuff, to form this mature messenger RNA. That then in the process of translation, this complex called the ribosome is going to travel around along it to make the protein. And it's going to piece together the amino acids through those peptide bonds in the order that's corresponding to like the sequence that they're traveling along. So it's read in these like 3-letter words called codons. So basically, and it goes, these are read like non-overlapping. So what it goes to one 3-letter, then another 3-letter, then another 3-letter. And at each 3 letters is corresponding to this anticodon in this transfer RNA or tRNA, which is also going to have the corresponding amino acid. And so what happens is that in the process of translation, this ribosome is going to travel along, and it's going to put in the amino acids corresponding to that sequence. So it's degenerate, which means that multiple codons can spell the same amino acid, but it's not ambiguous. So one codon can only spell one amino acid or stop, and it's not overlapping. So they're read like 3-letter, 3-letter, 3-letter. And so you can look at like a codon table, and you can see that there's basically this like universally agreed upon code that's used by all organisms for the most part. Where different 3-letter combinations are going to spell different amino acids, and these are going to be put in by those tRNAs. So when the cell is like, well, like through evolution and all of that stuff, when these proteins, like recipes are being made, they have this menu For most organisms, you have this menu where you have these 20 amino acids to choose from. We also talked about selenocysteine, and I'll get more into that in a bit. But these are like the 20 basic ones. And you also, so you have codons for making all 20 of those. Then you also have stop codons. So you also, so the methionine codon, AUG, is also the start codon. So if it's happening at the start of a protein, so it signals start, and if it's just like in the middle, then it's just the methionine like normal. But you also have to be able to tell the ribosome when to stop. And so what happens when you reach a stop codon is that a release factor is going to come in. So instead of a tRNA coming and binding, a release factor is going to come. And this is the little protein that's going to come and it's going to break this off in this termination. And so this is going to happen at these stop codons. And so we have these 3 stop codons that we use. But some organisms, they're able to, they have like suppressor, they've suppressed one of the stop codons. So instead of using it as a stop codon, they actually use it to encode an amino acid. And what happens in some of these like archaea and bacteria, so archaea are like these kind of bacteria-y like things. So they have some things in common with like prokaryotes, like bacteria, and that they're like single celled and they're have like don't have like some weird organelles and stuff, but they also have some things in common with eukaryotes, which are like plants and animals and stuff. So they don't really fit nicely in the tree, and there's a lot of evolutionary researchers and stuff trying to like classify where the heck do these guys fit in. But they're often, a lot of them are like extremophiles, and so they live in like really harsh environments. And there's some that are able to break down like products that we wouldn't, like most organisms can't. And these can end up generating like useful products for us to use. So there's some organisms like methanogens. So they're able to break down things like these weird things called like methylamines that can be breakdown products of other things, but basically they can make methane, but they can't break down methane. And so then we can use them to make methane, which then we can use as like a natural gas. So really cool stuff. And so they've been studied a lot in terms of their potential uses for like biotechnology stuff. But when they were studying these guys, they realized that the proteins, some of the proteins that they use to make, do this breakdown, actually they had a stop codon in the middle of the protein. And it was still making like a functional protein that was blowing past that stop codon. And what happened was it was putting in an amino acid that this amino acid that had never been, this amino acid turned out to be this molecule called pyrrolysine. And then some really cool experiments, I'll show you a little more later, they figured out that it was actually made out of lysines. But basically, in these organisms, the UAG, which normally spells stop in most organisms, is used instead to basically spell pyrolysine. They only very rarely use the UAG as a stop codon, and when they do want it to mean stop, they normally have like multiple stop codons in a row or some sort of backup system so that it actually stops. So basically, they've rewired things so that they're basically using this most of the time. So there's some like a lot of work into why they have this group and like what unique functions it's playing. For us, it's not, I guess, that interesting in terms of we wouldn't have like a use to put in like a pyrrolysine to introduce some cool function that we can then like visualize it or test various things. Not really, at least. Because this machinery that the cells use to incorporate this pyrrolysine, that is something that we could definitely take advantage of, and we can modify it in ways that'll make it so that it can put in a different amino acid. And this amino acid could be something that we're really interested in. So although we have all of this, like there's various functional groups, these basically the things that aren't carbon and hydrogen, things that are like kind of more interesting, things with like oxygen and nitrogens and things that are more reactive and can do cool things. We have all this variety, but sometimes we want we want more, we want more. And so especially if we want to introduce something into a protein that's going to make it different than a normal protein. So what if you could, for instance, introduce a group that was fluorescent, or a group that was photoactivatable? So basically it would be, you would have the cool part hidden. So the protein would be like inactive, and then you could shine light on it and it would break a bond, and then that would activate it. So there's all sorts of really cool stuff that you'd be able to do if you could incorporate one of these like unnatural amino acids. So there are ways that you could, there are definitely ways that you could modify amino acids. So we've talked about how there are like post-translational modifications that you could do to an amino acid. You could do things like, depending on the amino acid, you could do things like methylating it, acetylating it, so adding these like CH3 or these like carbonyl carbon double bonded to a methyl, like this acetyl group. You can phosphorylate some of them, this like phosphate group. With cysteines and lysines, you can use their like the nucleophilicity of their side chains to attach things to it, various things. But these require either the help of like an enzyme, so like a protein helper, or they require like some like chemical modification approach where you're doing things in like a test tube and adding something after the fact, after you've made the protein. And the problem is it can be really hard to alter exactly where you want to alter. So for example, if you're using just like some sort of reagent, some sort of chemical that's going to modify lysines, or cysteines, it's going to like add a fluorescent label to them. Well, it's going to do that to all of them, basically, all of them that it has access to, like all of them that at least that are like on the surface or whatever. And what if you just wanted to test a single one? Or if you use a kinase, it would do what the kinase wants to do. It wouldn't, you can't say like, put it precisely in on this residue. So residue is just like another name for an amino acid once they've linked together. So you can't say like, put it right there and only right there, not right there. And put it right there and not anywhere else. And so as a person who's been studying protein phosphorylation for like 5 years and trying to study this protein that has multiple phosphorylation sites, and I'm trying to tease them apart, it would be really awesome if I had a way to specifically control exactly where the phosphorylation was going to happen, and like different combinations and all sorts of different things. And this is one of the things that you could potentially do with unnatural amino acid incorporation, is incorporate the modified amino acids or versions that are kind of like mimicking them into the protein before it's made. And in order to do this, you would like, if you could specify exactly where you want them to be. So there are some methods for incorporating unnatural amino acids where basically what you do is you use like an analog of them that looks really, really similar. So it can tease the, it can trick the machinery into using it, but it's not, but so you're restricted to using something that's really, really similar so that it can trick the machinery. And in that case, what you're doing is it's going to get added wherever that amino acid, the normal version of the amino acid would normally get added. So we can do things like adding like nuclear isotopes, so like heavier versions of like carbon or nitrogen or hydrogen, if we want to do things like various mass spectrometry things, NMR, various ways in order to kind of like track things. So we can do things like that, or we can do things like add selenomethionine. So the version of methionine that has a selenium here instead of a sulfur. If we grow like the cells that are making our protein, if we give them selenomethionine, it'll incorporate selenomethionine where there's normally methionine. But in that case, you're replacing it everywhere because you're not changing the instructions for putting it in. In all of those cases, what you're doing is you're either in post-translational modifications, you're making it as it is and then trying to modify it where you want, or in the case of the this like analogs, you're just adding an analog that's going to replace all of them. If we want to change a site very specifically, we need to actually change the DNA code that's going to make the messenger RNA copy that's going to make the protein. And we have ways to change the code. much more on this sort of thing in other posts. But the basic idea is that we can put the genetic instructions for making a protein into cells and get them to make the protein. And so we can change that sequence in order to change the protein. And there are also ways you can change the sequence where it is actually sitting normally, like in the bacteria's own genome or whatever. But once you change that sequence, then you can change the amino acid that gets incorporated, because if you change the codon, now you can incorporate a different amino acid. But you're still restricted at this point to adding the things that the codons already know how to make. So the cells are restricted to using this set of like 20 amino acids. So the idea with using like unnatural amino acid incorporation in the sense of like expanding the genetic toolkit is that you need to either get the ribosome to do something really weird, like recognizing a 4-letter codon instead of a 3-letter codon, or doing things like replacing an amino acid, like replacing what normally spells one thing to get it to spell a different thing. And the common way that this is done is through suppressing one of the stop codons. So normally when the stop codon shows up, the release factor will bind and the protein will get cut off the ribosome until the protein will get stopped. So we have these 3 stop codons, but it turns out that there's great differences in the usages of these. So for example, if we look at E. coli, see that it uses UAA the most, like 64% of the time, UGA 29%, and UAG only 7% of the time. So you can do things, if you were to suppress the this amber stop codon, it especially because when you suppress it, you're not doing it like fully because there's still like competition from the release factors. Turns out you can do things like introduce unnatural amino acids, tRNAs that are with like coding for that amino acid to suppress the stop codon where you want it. And it's not like completely efficient because you also have those release factors and you could have it like misincorporated into other proteins and stuff. But it is commonly used and it is feasible. So this like amber suppression is a really common strategy that is used. So replacing this UAG, instead of spelling stop, in certain cases, you get it so that it spells a natural amino acid that you add. So in order to do this, you need to make a link between this unnatural amino acid and the codon that you want it to bind to. And so this means that you need to get a tRNA to recognize this amino acid, the unnatural amino acid, and to recognize this codon. The amino acids, when they're prepared to be added, it's done with the help of an enzyme called an amino acyl tRNA synthetase, or an AARS. So an acyl is just, you have a carbonyl, so it's double bonded to an O, bonded to some other group. And so what's happening here is that this amino acid is acting, so this part is like an acyl. And so the tRNA gets prepared with the amino acid, but with this amino acyl tRNA synthetase, and it's going to do this in 2 steps. And it's going to take ATP. So it requires some like energy money. So ATP, I think about it as like energy money, because you can think of these negatively charged phosphate groups all held close together as being kind of like a clamp spring. So if you were to cut this apart, you'd release energy. And so then this also makes this more reactive in the following steps so that it can be added to the tRNA. And this energy investment is worth it because basically when it gets to the ribosome, what the ribosome's looking at is it's looking at this codon, this anticodon, which is going to match the codon. And so the ribosome's not going to care what's like attached to this tRNA. The ribosome's just going to care what's this anticodon. So you want to make sure that you have the right amino acid loaded onto this tRNA. And so how this is done is that you need this AARS to be able to recognize the right amino acid and the right transfer RNA. And so what's going to happen is that the ATP, the amino acid, is going to be adenylated. So it's going to have like this adenosine phosphate group added on. And then this is going to then make this vulnerable to attack by this tRNA, which is going to get you this amino acyl tRNA product and AMP. And then this amino acyl tRNA product, it's going to go in translation. So when its corresponding codon shows up in the ribosome, then it's going to come with the help of an elongation factor. It's going to come and it's going to bind. The growing chain is then going to get transferred to this incoming amino acid. Then elongation factors are going to help shift things over. The next amino acid is going to come that corresponds to the next codon, and continue and continue and continue. And then what happens normally is that when one of these stop codons shows up, then a release factor is going to combine. So this little protein is going to combine and it's going to cut this off. If we want to take one of those stop codons and change it into spelling an amino acid, we are going to need to get there basically before the release factor. And we're going to need to have a something that competes really well. And so we need this tRNA. We're going to have a tRNA that's going to actually specifically match the sequence, specifically match that stop codon in its anticodon. And so what's going to happen is there's a couple examples in nature already of this. And so yesterday we looked at the case of selenocysteine. So selenocysteine is actually used by like our cells and stuff, whereas pyrrolysine isn't. But in selenoproteins, what happens is that you have this, so selenocysteine is like cysteine, but it has a selenium instead of a sulfur. And so yeah, it's made from serine to form selenocysteine, which then gets added into proteins. And how it does it is it has this like special element in the RNA, this like cassettes structure in the RNA that's going to bind to these like helpers, and it's going to kind of keep this selenocysteine loaded tRNA really close by so that it can kind of sneak in there before the release factor will bind. So in the cells normally, however, this UGA is used as a stop codon a lot of the time. However, in the case of pyrrolysine, this other amino acid that's found in some like archaea and bacteria, what it's going to do is, so it uses a different stop codon. It uses UAG, which is that amber stop codon that isn't used that much. And so what it does is basically it almost never uses it as a stop codon, and instead it just uses it as this spelling pyrrolysine. And so because it doesn't use this as a stop codon really, then it doesn't have to use as many tricks in order to get in and sneak in there. So in order to get it in there, you need to be able to make it. You need to be able to make this pyrrolysine. You need to be able to make a tRNA that'll bind to, that'll like transfer the pyrrolysine that has the complementary anticodon. And then you need your tRNA synthetase that's going to be able to load that tRNA with the pyrrolysine that you just made. And so these scientists were able to figure out that the genes for doing all of this in this archaea, these genes for doing all of this were basically clustered in this one little region. There were 3 genes that were needed to make it. We'll look at these in a second. This pile B, pile C, pile D, and then there were 2 that were needed in order to stick it in proteins. The one for making the tRNA, the pile T, and then the one for the synthetase, the pile S. So pile S is the gene, and then the product is this enzyme, pyrRS, pyrrolysyl tRNA synthetase. So when these organisms make it, so they need to make it to put it in. But if we want to use this system, so because you have it in this like cassette like manner where basically you have it all together that you can then insert into something else to get those like other cells to make this, you can actually, if you don't need the pyrrolysine, you only need these two genes. But if you don't make the pyrrolysine, then these guys have like nothing to do. But the trick is if you can change these, you can make mutations in these that'll allow it to make, to incorporate different amino acids, which then you can supply like exogenously. So basically you, instead of making the cells make it themselves, you give them something that you want them to incorporate. And you engineer this like tRNA synthetase to be able to recognize that thing that you added and get it loaded onto the tRNA. So there are a bunch of different strategies that involve with like directed evolution and stuff. So in labs, basically they can make a bunch of mutations to it and then test the functions because you need to make sure that it's orthogonal to what's already in the cells. So basically orthogonal means that you can, it's going to be like separate from what's already in this, like outside of what's normally in the cells. And in this case, what we really care about is that the tRNA synthetase is not going, this amino acid that you add isn't going to get loaded onto any other tRNAs, just this one tRNA. And you need that this one tRNA isn't going to get loaded with any other amino acids. So you have those two things that you need to worry about. And so you can make changes in both of those and then see like, is it getting loaded with just the one we want? Is it getting loaded, is this getting loaded into other things? And so there's a bunch of different strategies that scientists can take in order to evolve these proteins to be used to incorporate different amino acids. So there are literally like hundreds of different amino acids that have been incorporated. So these are just the ones that have been like incorporated into mammalian cells, and they can do all these things just by making differences. So you can see they're making differences in the genes for these. See, they're making differences this in the genes for like this tRNA synthetase and stuff. So there's all these different modifications, and there's really cool stuff that you can do with like caging and stuff. So this is like photo activation and photo deactivation, and so you can like make things switch on and off, all sorts of really cool stuff. And so I'll post links to this review paper, and like here's another one, really good. But of course, the inspiration for all of this is coming from this work on discovering pyrrolysine. And and then looking into how that pyrrolysine is made. And so let's just look at this in a little more detail. So making pyrrolysine involves these three genes for these three enzymes. So pyrrolysine is made from lysine. So basically it's made from two lysines that form pyrrolysine. And how this works is that this PAL-B is going to do this, like it's a mutase. And so basically it's going to change the structural arrangement of this first lysine. So it's just going to change how things are linked up. And so it requires a SAM cofactor, so S-adenosylmethionine. So a cofactor is something like, it's like a enzyme's like helper or like sidekick. So it's another molecule that the enzyme binds to and uses to help it work. So it's like they're modifying them. See, they need like extra functionalities as well. But in this case, it's S-adenosylmethionine. So we talked about this guy a lot when we were talking about methionine and methylation, but there we were talking about how it was used to move a methyl group. That's not what we're talking about here. S-adenosylmethionine, or Adomet, it actually can play other roles, and it can play roles in like radical reactions. And so radical is where you have like a free electron, and these are really reactive. So what this is going to do is that this PAL-B with SAM is going, so SAM is going to help this PAL-B basically steal a hydrogen with its electron. And so this is going to leave you with a lone electron. And so this is going to be an unstable radical intermediate. And then this is kind of going to attack, but it's going to attack here. And so you get this 3-methyl-ornithine compound. This is then going to join with a second lysine molecule with the help of pyrrolysine. And this step is going to require ATP. What's going to happen is, this is going to link up in this way. This lysine, so this like end lysine, which we talked about when we talked about lysine, how this guy's is really like nucleophilic and can like attack and react to things. So nucleophile is something that like has more electrons or electron density than it wants, so it seeks out an electrophile, something that wants electrons. So much more on that in other posts. But basically, you can get this nitrogen with the help of the enzyme to get this nitrogen to attack, form this kind of like amide bond here, like the bonds connecting proteins. But in this case, what's happening is you're connecting the end of this lysine, so not the alpha amino group, but this epsilon amino group, to this carboxyl, what was the carboxyl group of the other, of this weird modified amino acid, this 3-methyl-ornithine. So when you do this, now what's going to happen is it's going to get like oxidatively deaminated. So deaminated, you're going to remove this amine group and you're going to oxidize it. And this is going to be done with this enzyme PALD. So basically you're going to cut off this amine group and this is going to get converted into an aldehyde. And then what's going to happen now is you're done with the enzyme catalyzed stuff, but this guy's unstable and it's going to spontaneously cyclize. So just like we saw before when we saw like a nitrogen be attacky, this nitrogen can basically attack this, this aldehyde, and this is going to like ringify this and kick out water. And so this is what we call pyrolysine. The name comes because this is a pyrrole group. This ringy thing is called a pyrrole. And so this is like a methylated pyrrole. So you have this methyl group. And then once you make this pyrolysine, then you can sneak it into proteins. And they don't even need to be that sneaky because the cells are normally using it. So this pyrolysine was discovered in like 2002, Joseph Krzyzewski and Michael Chan. And so basically they were studying these methanogens that like have these like methylation activities. It's kind of confusing that these little microbes do. So I'm not going to go into that, but basically they found that they were making this protein with the stop codon in the sequence. And then they were trying to figure out how it all worked. And so they found that there was this cluster of genes that you needed to do this. But they were still like not exactly sure what it was that was actually getting added. So basically they tried using like mass spectrometry where you can like look at the size of various fragments and stuff, but they couldn't tell what was happening there, especially because you can have modifications like during the fragmentation, you could have fragmentation that you don't want and stuff. So they couldn't really tell what was being added. They just knew that something was getting added in the place of this normal stop codon. And so they collaborated, the Krzyzewski lab collaborated with Michael Chan in his lab. They were actually able to solve the structure of this protein that had this pyrolysine incorporated, and then they were able to figure out the structure and figure out what pyrolysine actually was. They were able to show that you could then stick this, they were able to show that you could take basically those 5 genes and like stick it into other cells and get the cells to make and incorporate pyrolysine. So that was all you needed was this like cassette. And so this together with that like opened the doors for unnatural amino acid incorporation and various things like that. but they still weren't exactly sure how it was working, that it was all happening, and like where the pyrrolysine precursors were coming from. So there was a lot of like controversy about like where the parts were coming from that were used to make this pyrrolysine. And so then in 2011, they showed that it was actually coming just from lysine. So other people had thought maybe it was coming from like, it was like ornithine or that sort of thing involved. But then they have this, they basically use labeled. So the actual article is here. And then there's also a good like editorial summary type thing explaining it, as well as the work leading up to it. But basically they use like isotopic labeling to label the lysine. And then they were able to then analyze the protein using mass spectrometry. So because you have like the lysine, you have the labeled lysine, it'll be heavier. And basically they found that the pyrrolysine in the protein was all heavy, like all the parts were heavy that were coming. So it was coming from just from lysine. And so then they also showed that like ornithine can be used, and that, but it makes this, so they showed that ornithine like could be used to make like this altered form of pyrrolysine. So this version without the methyl group. And this was also kind of like a, oh, this guy's kind of a little promiscuous. Like we can get this tRNA synthetase and this tRNA to add to use slightly different substrates. And so then this kind of opened the door for doing things for even more, for incorporating unnatural amino acids using this system. So their initial findings kind of like opened the door for using this sort of suppression, and then this is going to help open the door for incorporating a variety of different amino acids. These discoveries were really important because it would be like super duper hard if you had to try to genetically engineer these proteins from scratch. Like just, if you were to just like try to take a computer and design something that would add a various thing. But by having this work done, by having this tRNA synthetase and this tRNA, you could then just modify based on that. You have like a starting point, and then you can do a bunch of screening in order to find things that work, things that don't work. So various really, really cool things. And so I think the unnatural amino acid incorporation is just like really awesome. And it's really built on the work of on this pyrrolysine work in large part from the Krasicki lab from Ohio State. So really, really cool stuff. And I look forward to see what happens in the future. So going back to comparing pyrrolysine to selenocysteine, basically, so selenocysteine is going to use a codon that's normally used as a stop codon, like most of the time used as a stop codon, like almost all of the time used as a stop codon, and it's used a lot as a stop codon. So it has to be tricky. It has to use the special tricks of like having all of these other factors help it and have this element in the RNA. And also, so this would be like a cis-acting element because it's on like the same thing basically as the stop codon. And then these would be like trans-acting elements, so like external factors. And so all of this needs to come together in order to sneak this guy in. But in the case of pyrrolysine, because they don't normally use this guy as a stop codon, you are able to use a tRNA like exclusively basically in order to incorporate this pyrrolysine. Another important difference is that with the selenocysteine, it's actually getting made from serine after it's loaded. So that the serial tRNA synthetase is actually going to put a serine onto the selenocysteine's tRNA, and then this is going to get modified to have the phosphates. It's going to get phosphorylated, and that's going to get swapped out for selenium. And so selenium is like your cells keep tight control over it. And so you have to go through a bunch of special steps in order to really controllably add it. And then you have to go all these steps to sneak it in. Whereas with pyrrolysine, basically you can just sneak it in as is. And the pyrrolysine is going to be made from lysines before it gets loaded onto the tRNA. So it's loaded on as pyrrolysine. It's not loaded on as something else and then modified. And so some people say like, well, technically then this is the 21st and not the 22nd amino acid, but this was discovered first. But some people say because it's not getting loaded with it. Yeah, but it's all just like semantics and terminology and stuff. But anyway, this is typically regarded as the 22nd amino acid, and this one will be the 21st. And the 22nd amino acid is like the pathway to countless amino acids